All right, so back to chapter 1. We know microorganisms, just to review for the recording purposes, okay, we know microorganisms are too small to be seen with the naked eye. Acellular microbes are going to be viruses and prions. Prions are our misfolded proteins associated with the brain disease of spongiform encephalopathy because we're poking holes in the brain causing disease. Eukaryote is a term that is interchangeable with prokaryotes, and we believe they're the first organisms on the planet. Number one, because they're the simpler versions of the cells. Number two, because we actually have evidence of the organelles in the eukaryotic cells. True nucleus is what eukaryote means. The eukaryotic cells seem to have organelles that are related to, the eukaryotes, which is quite wild. And we'll learn more about that in chapter 5. And also, it seems that our atmosphere on Earth used to exist without any oxygen at all. And it's thanks to microbes that learned to live in that oxygen-free environment that they made oxygen as a waste product. And that is really what led to being able to support more forms of life that depended on oxygen to live. And so it's thanks to that that we even have oxygen on the planet at all. Plants do make quite a lot of oxygen, but the primary source of oxygen on our planet is going to be things like algae, which are actually protista, which you'll learn in chapter 5. So, but yes, so that's the gist of our microbes that we have going on there. A pathogen is a term for any microorganism that causes disease-causing contagious, disease-causing agent. This will be any of our microbes that cause disease. Now we have to remember there's quite a lot of microbes that don't cause disease. So remember that not all microorganisms are pathogens, and even not all microorganisms could be pathogens. In fact, there's going to be microbes that couldn't even survive in the human body if they wanted to. So there's plenty of microbes out there that are not pathogens. We're going to talk mostly about the pathogens in this course once we start getting into things. Then it led us to talking about germ theory and the causes of disease. Germ theory is the idea that these contagious diseases that seem to be spreading from person to person must be caused by a germ. We call those germs pathogens now, but they did not come from spontaneous generation, was the original belief at the time. Spontaneous generation was the idea that if something was left alone long enough, life would come from that. And so that's just not how it works. It just was they couldn't see the life. It's just too small for them to see with their naked eyes. And so as it turns out, germ theory was created thanks to Louis Pasteur and Robert Koch. They did a lot of research that led into those investigations and proved that contagious diseases were in fact caused by what they called germs at the time, but truly we call them microorganisms now, more commonly associated with pathogens. And it was Robert Koch that came up with these steps to investigate the causes of disease, what we would call etiological agents. And we'll talk more about etiological agents and how diseases are diagnosed and how we figure out the causes of diseases in later chapters. That's going to be Unit 2, Unit 3 thing. So we're not going to get into it yet, but we'll learn more about Robert Koch and his contributions to science a little bit later on. And then we have aseptic technique, which we have learned about in lab today. If you are in my evening class, we will be doing that in lab here in just a couple of hours. But aseptic technique is the idea of preventing contamination. So we learned that in lab. We want to prevent contaminating our sample with the environment, and we want to prevent contaminating the environment with our sample. And this is true anytime that we're dealing with any sort of pathogenic organism. We want to be sure that, or potentially pathogenic organisms, we want to be sure they're staying where they are and not, getting out of there. And definitely, even when we're talking about treating things in medicine, we don't want to spread that disease to other patients. And so aseptic technique is actually something that's practiced continuously throughout the medical community. And it was Joseph Lister who came up with this concept of basically disinfecting his operating theaters. He used phenol, also known as carbolic acid, to basically spray down his operating theaters to clean them. And he also was one to start practicing hand washing before his procedures as well. So his patients did considerably better, and his practices were taken up by others and eventually, of course, perfected into what we know today in surgical techniques with that, sterile field and everything like that. Another topic that was discussed in chapter 1 was taxonomy, which is just about the most boring thing that I could probably talk about because I feel like it's just terms for the sake of terms. But the idea of taxonomy is basically to organize organisms either based on their genetic relatedness or based on like feature characteristics. And so we can group organisms into these categories based on their traits, their genetics, and all sorts of stuff like that. The most general group is domain, and you do need to know the 3 domains. The 3 domains are eukarya, bacteria, and archaea. As it turns out, if you look in a microscope at an archaea, they look exactly like bacteria. They live in more extreme environments than bacteria, typically, not always, but typically. And they rarely are associated with disease, but there are some that are associated with disease. I think we talked about one in one of my sections, which was the Methanobrevibacter oralis, which is associated with severe periodontal disease, like gum disease, tooth decay, and things like that. And if you have that archaea, then you are more likely to have severe presentation of that disease. Well, you don't see a lot of archaea other than that in diseases. So we do not talk about them much. Their cell walls, their cell membranes are made up of wildly different compounds than what we think of with a typical cell. One thing you're going to learn in chapters 4 and 5 is that cell membranes are made up of phospholipids. This is actually pretty common. You've probably talked about this in chemistry, or if you've had any biology at all. Your cell membranes are made of a lipid bilayer made up of phospholipids. Not archaea, not archaea. Archaea have like some sort of weird, I'm sorry, the way I'm sitting is incredibly uncomfortable. Okay. Archaea have some sort of wild like hydrocarbon thing going on with them that like it's completely unrelated to the bacterial cells or the eukaryotic cells. And they're more genetically related to eukaryotes than they are to bacteria. But they have a lot of other things that are common with bacteria. So they're kind of in between, but they're so wildly different. They are their own thing. So archaea are their own thing. They are their own thing, just like we are different from bacteria. They're that different. So they are wildly on their own deal. Let them be. So our 3 domains are eukarya, bacteria, and archaea. In the eukarya group and in the bacteria group, we basically subdivide everything down the same way. Okay, so after domain, you belong to kingdoms that are within each of those domains. And then within the kingdoms, you belong to a phylum within the kingdoms. And you belong to a class within that phylum. And you belong to an order within that class, a family within that order, and then a genus within that family. And when we write an organism's name, where their species name, we write in binomial nomenclature. If you're writing it by hand, you would write it like Staphylococcus aureus. It doesn't like me right now. And you would underline it if you're writing it by hand. If you're typing it, you would italicize it instead of underlining it. Okay. So there are rules there, and you will be tested on those rules on the exam. You've been warned. Okay. You also have to capitalize the first word in the binomial being 2 names, right? So you have to capitalize the first part of the 2 names and lowercase the second part. If you're typing it, that would all be italicized, which is what the test will have on it, just saying. Okay. So you want to be sure that all those boxes are checked. Is the first word capitalized? Is the second word lowercase? Is the whole thing italicized? Or if handwritten, is it underlined? And so that's the proper way to follow binomial nomenclature. I'm really not going to test you on it unless I'm telling you that I'm going to test you on it. So on the exam, you will have a question that will require that. on your lab reports and on your presentations about your diseases, you should follow those rules. But pretty much outside of that, I'm not going to be super strict about it. So just know it in those contexts, okay, and follow it in those very specific, more formal contexts where appropriate. But otherwise, if you're writing up a concept map or something, I'm not going to be as picky about it in those situations, okay? But that is the gist of that one. Any questions about chapter 1 stuff? I feel like chapter 1's pretty straightforward, right? I think most of us agree. It's a short little blip, especially how I cut it down for the presentation. So let's move on to chapter 3. It has a little more to it, but still, if you've gotten to chapter 4, then you know chapters 1 and 3 and even 2 aren't really that bad. Chapter 4 is where we start getting some of the meat. I'm telling you, if we can get unit 1, if we can get unit 1 under our belt and really under our belt and hold on to that info, it's going to make unit 2 so much easier. And units 2 and 3 are the challenging units. All right, so let's move on to chapter 3. This was the tools of microbiology. We have first the 5 I's. So the 5 I's, our first one was inoculate. Our second one was incubate. Our third one was isolate. Our fourth one is inspection. And our fifth one was identify. We'll just put inspect, but whatever. So there's the 5 I's and they mean something. So don't just memorize the I's. Know what each of those I's represents. Inoculating represents putting the sample onto the media. Incubating means growing it at its ideal conditions. Isolate means separating it from any contaminants or mixed cultures. Inspection means you're doing testing on it to gain data about it. And identify is actually using that data to confirm the identity of the organism. So each of those 5 I's has a reason and has a purpose whenever we're investigating a microbe. Next is differential and selective media. Yes, there were more media, so we also have some other stuff. Differential, selective, and enriched. We're going to talk more about enriched in a moment. There were other medias that we talked about, like the complex media and the defined media. The complex media I've talked about in the beginning of this, but if you guys weren't there, complex media being basically the type of media that you can't actually determine the exact molecular makeup of. When we add 5% sheep's blood, can you tell me the exact molecular makeup of that sheep's blood? Probably not. So that's complex media. But defined media, we've defined every single thing in it down to the molecular level. We know the moles of how much of each ingredient there was in there. If you guys remember moles, this is probably the last time I'll bring up moles in this class, but you get what I'm getting at there. So then we had talked about selective and differential media. Selective media is the type of media that selects for something. So usually you're favoring something, you're selecting for something while also selecting against another thing. I gave one example at the beginning of the lab or at the beginning of the review. I'm going to give another one now. So one example you can think of with selective media, I actually will go over both. I forgot that I wasn't recording at the beginning of the review. So the example I gave in the beginning of the review, when a student asked a question was about like selective and differential media. And I gave the example of mannitol salts agar. So mannitol salts agar is both selective and differential. And you will learn that later on in the course. Right now for chapter 3, I don't need you to tell me about McConkey agar or mannitol salts agar, but I do need you to tell me about what selective and differential media is, the broader idea there. But an example of a selective media, and if I were going to ask you a question about it on the Unit 1 test, I would tell you about the media. But if the media has something in it that inhibits the growth of certain organisms, that's a selective media. Because some things will grow on it and some things won't. So mannitol salt agar is like that. It has sodium chloride in it, and sodium chloride, salt. We already know if you pack cells in salt, what happens to the cells? They shrink up. The water comes out and goes into the salt, right? And it shrinks up the cells. Well, gram-positive bacteria, they have thick cell walls, which you will learn about in chapter 4, so no worries, it is coming. Like I keep saying it is, but it really is coming. So chapter 4, you learn about bacteria, gram-positive bacteria having these thick cell walls. Gram-negative bacteria don't have the thick cells. They have a very thin cell wall, and honestly, it's not doing a lot for them. So gram-positives, they have that thick cell wall that helps them survive better because it's holding the structure of those cells, even in the presence of the salt. And so they don't shrink. They're able to withstand the salt. And that's why most of the bacteria that grow on your skin, like Staphylococcus aureus, Staphylococcus epidermidis, and other bacteria that will be found on the skin or like kind of our outer areas that can get dried out, that can get salty and things like that, those tend to be gram-positive bacteria. The bacteria that live in your gut tend to be gram-negative because they can withstand certain things that the gram-positives can't. Now, this is not a hard and fast always rule. There are, of course, exceptions to these rules. But overall, those structures play a pretty big role in where those organisms can survive. And we can actually use that to our advantage when we're culturing something. So if you have a patient that has a skin infection, like we had in our lecture activity, where Liam had crusted sores on his face, the little kid Liam, right? He had the crusted sores on his face. So we have every reason to believe if it's growing on somebody's face like that, it's something that can survive dry and salty environments. It's probably going to be a gram-positive organism. We can assume that for sure. And then we can use media that selects for gram-positive, but we can also culture it on the gram-negative favoring media just to rule it out at the same time. So that even tells us something about that organism without even having to do the stains. So there's that advantage with the media. There's also the differential media. So moving on from selective, selects for some things to grow and not others. Differential media tells you the difference. So we said mannitol salts agar is both selective and differential. It's selective because only things that can withstand salt can grow on it. So it's selective. But once you grow on it, if you're gram-positive, you grow on the mannitol salts agar. Some things on mannitol salts agar are going to turn it a different color. The media will literally turn a different color because those organisms can ferment the sugar mannitol that's in the media. So not all organisms will do it. And when we ferment things, one of the byproducts can be acid. And that shift, that decrease in the pH will actually cause the pH indicator in that media to turn colors. And then that would be an indication of a difference. So differential media allows you to see differences. Selective media allows you to select for what will grow and what won't. And you absolutely can have medias that are both selective and differential at the same time, just like mannitol salts agar or McConkey agar. There's other kinds of media, the media that has special growth factors in it. So the first thing to understand what this media is called enriched media, but in order to understand enriched media, we have to understand what a growth factor is. And we did some of the labs, we talked about this. Growth factors are things that are required to be supplied in the diet. So we have this as human beings. We have to have vitamin C supplied to us. We have to have vitamin B12 supplied to us. Our bodies can't make those things. And so we have to have them supplied. We even have some amino acids like lysine. Our body can't make lysine. We have to have it supplied in our diet, but our body needs lysine to build our proteins. And so you can actually die without these growth factors. And in fact, some bacteria are like that too. We often call our growth factors vitamins, not always, but that was a more colloquial term that now is really something that we can apply to just general growth factor. So growth factor is something that is required in the diet that the organism can't make for itself. And some of our bacteria need that. Whenever you are an organism that needs a growth factor, like humans who need to be supplied with vitamin C or B12, then those organisms are called fastidious. So human beings are fastidious because they require growth factors in their diet. Any media or food that the organism will eat to get the growth factor the media that we supply it that has the growth factor in it, we call that enriched media. In humans, you buy enriched foods all the time, whether it's your cereal or your bread, and it will say literally enriched. It means the same thing. It means it has vitamins added to it, like riboflavin, which is just a B vitamin, by the way, has these things added to it in order to add in those growth factors to be sure that you're getting them in their diet. And so our bread and things like that are enriched, just like some of our media might be enriched. So blood agar is an example of an enriched media. We add blood in it because some organisms won't grow without the blood. So that's a growth factor there. So that's an example there. All right. Chapter 3 also talked about microscopes, and hopefully this stuff was a little straightforward for you guys. We do need to know how to calculate our magnifications. If you have a microscope and we'll go over microscopes next week, but you basically have this setup where you have this little rotating guy and you got some lenses coming off of the rotating guy, and this lens is looking at the slide that's sitting on the stage here. And that stage has a condenser through which the light will shine from your light source. Here's your light source here. This is the light source down here. Here's your light bulb. Quit. And then here is your microscope. Okay, cool. There's your microscope. We love it. It's such a good picture, isn't it? So anyways, the light comes from your light bulb down here out through the condenser. There is an E in there. I did write it. It just didn't pick it up. This is the stage. Well, right, light source, just to be sure. This is your slide. And the light passes through the slide, through the condenser, and the little hole in the condenser that it's passing through, we call it the iris. And you can control how open or closed the iris is with something called the diaphragm. We'll learn about that in the lab. But the light comes through the condenser, shines through your slide, and it hits. It just does not want to let me draw a squiggle. Okay. It hits your first lens. So these are objective lenses. Once it hits that first lens, at this star here, it creates an image. The lens, whatever image is from your slide, gets magnified by your lens, and it creates the first image. The first image that we're making, because that's the first place the light is hitting, okay? This first image, we call it the real image. It's the first one. Once it shines in there, it'll travel up through the microscope, and through some mirrors, it'll shine off and reflect off some mirrors, some different ways. This is inside of the actual microscope. And then you would be able to look at it in an eyepiece. We'll just draw the eyepiece out here, whatever. There's another lens here. Eyepiece is usually 10x, and then your objective lens is whatever it says on it. It might be 10x, it might be 40x, and the ones we'll be using in our lab will work up to 100x, right? And you just multiply whatever your eyepiece power is times whatever your objective lens power is to get the total magnification. So in this case that I have drawn over here, our eyepiece, I told you up here, was 10x. Our objective lens, where we're shining through right here, that's also 10x. So overall, the image that you're looking at through the eyepiece is 100 times what the actual cell would be in that picture. So when we use the 100x lens, you're actually at 1,000 times, because the eyepiece multiplies it another 10 times. Okay, there's another lens in here that creates a second image. So we'll put a little star here. Keep the lens, please stop erasing it there. It's okay. This is our second image, because we're passing through another lens that's changing the image. And so because we're changing what the real image was, this is now the virtual image. Okay. Can we agree? I hope that my reviews aren't all going to be like this. I don't know what it is. My legs are really sore. I had Pilates yesterday, and I don't know, that lady worked me out. So my legs are really sore. It's just not comfortable for me sitting in this chair right now. Anyways, we can agree that air has a different density than glass, right? So, I mean, I can do this in air. Can you do this if you're encased in glass? I mean, literally encased in it. Of course not. So it's different density, right? So light has that same reaction. When light passes through glass and then hits air, that causes refraction. And what you need to understand about refraction is that that means scattering of the light rays. And so instead of your light that hits your image and the light that as it hits its image, creates the image, scattering all over the place once it hits the air. Now we can keep it nice and tight and contained in what field you're looking at by adding oil, which is much closer in density to glass than air is. So that's the point of oil immersion, to reduce that light scatter, also called refraction. When we're preparing our specimens, we often heat fix, which you will definitely be doing on Tuesday or Monday or Tuesday, depending on what class you're in, okay? Monday or Tuesday, lab 2, we will be doing heat fixing and we will be doing a simple stain, okay? So the whole point of heat fixing, it has some purposes, okay? The main purposes are, yes, to kill the organism, as well as to adhere it to the slide. Stop. It's not me doing that, I promise. It is the pen. All right, so it's either to kill the organism, adhere it to the slide, but it also fixes everything in the cell in place when we do that. Heat fixing is a little tricky. You don't want to burn the specimen because that actually degrades the molecular makeup of the specimen. So the idea is just to kill it, but not incinerate it, not oxidize it with heat, not burn it away. And so that can be a little tricky for some people in the beginning. Don't worry, you'll get plenty of opportunity to work on that. Yes, this will be posted. Absolutely. No worries. So the whole point of that heat fixing is to prepare it for the staining. So we will fix it onto the slide and it gets adhered there. And we're going to be doing a lot of rinsing on them in a lot of our differential stains. Most of our stains will be staining the cell. And if you guys know, and you will learn in chapters chapter 2 really covers it. It's chapter 2, the chemistry chapter. A phospholipid looks kind of like this, where that head is the phosphate group. And I don't know if you guys know this, but oxygens, they are really, really electronegative. And what electronegative means, as it implies, they hog electrons. So that head is very negatively charged, but these tails are hydrophobic. Chapter 2 covers this, but I'm bringing it up for a reason. Your lipid bilayer has a whole bunch of these heads with the tails on the inside, and it hides the tails on the inside. And it's like this the whole way around the lipid bilayer. Don't, Lauren. Okay, there we go. All right. You get it, right? So all of these negative heads are on the outside. So your cell has a membrane around it with all these negatively charged, and all cells have this negatively charged layer around them. So positive stains have a positive charge that is attracted to that negative. So positive stains stain the cells, whereas negative stains have a negative charge repelled by the negative surface of your cells. And so negative stains with a negative charge, they, stop it, pen, stain the background. So most of the stains that we're going to be working with are positive stains, but we absolutely will be working with negative stains, and you absolutely will get tested over which one is which, okay? There's a few different kinds of stains that we will definitely be doing in the lab, and you will learn more about these. But the Gram stain tells us the difference between cell envelopes. This is the cell outside. What's on the outside of things is what we call the envelope. So this tells us the difference between cells outsides, okay? The acid fast stain is used to see if you have a special wax on the outside, and chapter 4 goes over these in detail. All right. The endospore stain is used to see if you have structures called endospores, which are hardy and resistant They also are so resistant that they're resistant to staining. So we have to use a special procedure to stain them. But luckily, once we get the stain inside of the endospores, regular cells rinse free easily. They don't hold on to the stain, but those endospores will hold on to it for dear life. And so we can actually tell the difference to see if they hold on to the stain or not. That means they have endospores in them. And we will do an endospore stain. That's lab 7. Capsules, these are protective coatings, which you'll learn about in chapter 4. These are sugar or protein-based coats that really are protective as well as for adhesion. You can imagine that if you had a sugary, sticky, like honey stuck all over your surface, it's hard for things to grab onto you, but it also helps you stick to the things that you want to stick to. And that's the idea with the capsules. All right. Any questions about chapter 3? We're going to move on to the most missed topics for the week. For chapter 1, I think there's only one topic that we'll be going over, and that is the germ theory thing. So we're going to just solidify that. All right. The idea of germ theory, it clashed with what those current thoughts were at the time of spontaneous generation. It meant that infection was caused by something we couldn't see, but it was already there. It was there in our environments. And Louis Pasteur was the one that developed that flask experiment where he had an open flask with broth in it, and then he had a flask that had that twisty neck thing on it. I mean, this is really over exaggerated with how it looked. That let in air, but it didn't let in microbes. And then nothing grew in here, but it did grow in the one that was open. And he's like, clearly, if media just spontaneously generated things, it would have grown in both of the flasks. But clearly, we're letting something in into that first flask. And those we call germs. And he didn't really have a good understanding of what germs were or how to study them at the time, but he was right. So that is Louis Pasteur. And yes, you will get tested on the old guy names. If I bring them up in a lecture, you do need to know them. Okay. Here's some questions. Who developed and tested germ theory? So that was Pasteur. Remember Lister? He did the aseptic techniques in the operating rooms. Edward Jenner, we haven't really talked about him. I mean, I've talked about him like randomly, but he's the guy that did the smallpox vaccine, the first ever vaccine ever. The whole reason why vaccines are called vaccines. And of course, Einstein is theory of relativity and things like this is not related to this stuff. So this is Pasteur. So you notice how I answer this question and I don't just jump to Pasteur being the right answer. This is key because this was an easy enough question. But when you guys are taking my test or the nursing or whatever test you take after my course, sometimes the other answers might be more right than the one that you want to pick initially. So just make sure you're actually ruling out the wrong answers effectively. Okay, that's what I try to do on these reviews. Plague doctors stuffed the nose of those masks with herbs and flowers to ward off bad smells they thought were causing the spread of plague. Why was this ineffective? Well, this comes back to the germ theory, obviously, because that's what we're covering here. But would the herbs have worked if the herbs were antibacterial? No, because there's other reasons why there's bad smells. Rotting bodies might have viruses in them that you can contract, and those viruses have nothing to do with an antibacterial effect of an herb, right? Also, that's not really how that stuff works. Is having an antibacterial herb going to filter out? This is just not a very practical kind of a way of approaching this. Smells do not transmit illness. We can say yes to this. Let's see if there's any better answers than that, though. Did the flowers block them from seeing their patients clearly to treat them effectively? I mean, I have no idea. That's not why they were ineffective at stopping plague, right? They might have had that issue, but that doesn't answer the question. All right, were they ineffective because the contaminated herbs were being transported from home to home? We don't have any evidence that that was the issue, but we do think that the smells that they were trying to protect from were not what was transmitting the illness. In fact, if you were a plague doctor and you're blocking the smells in your mask, are you actually being protected from the fleas that were biting these people that were transmitting the plague? They didn't even think about that. So that's the issue there, right? It's not assessing the actual way that that disease was transmitted, which was through flea bites. That's how it was transmitted. All right, moving on. We have another one. This is something too. Is where are eukaryotes and eukaryotes, how they kind of are related to each other. Cell evolution. So eukaryotes, they lack a nucleus. A means without. Again, prokaryote, pro means before. And I don't care if you say eukaryote or prokaryote, your choice, as long as I know what you're talking about on the test. All right. Eukaryotes lack a nucleus, as well as other defined cell structures, which we call organelles. They don't have mitochondria. They don't have a nucleus. They don't have endoplasmic reticulum. They don't have a Golgi apparatus. They don't have vacuoles. They don't have chloroplasts. They don't have any of those things. They're literally just a membrane around some goo. And the stuff happens, the same exact stuff happens in the goo, but it's just not compartmentalized like a eukaryotic cell. So these seem to be more primitive. They likely came first in evolution. And there seems to be evidence that supports that, which you'll talk about in chapter 5. It's believed that mitochondria evolved from a bacterial symbiont. And a symbiont means that two organisms depend on one another to live. They don't have like transient interactions like you feed your dog. Your dog could find another place to eat from if you weren't feeding them, if it was let out in the wild. But a symbiosis means that we actually depend on for living. And so we think that there was a bacterial symbiont that became a mitochondria, okay? If you can't have eukaryotic cells without mitochondria, and you can't, that's how our eukaryotic cells get energy. Our mitochondria make that energy for them. Then what can you infer? Did bacteria come from the eukaryotes? We probably had to have bacteria first. We couldn't have had eukaryotes without the mitochondria. And we think the mitochondria came from the bacteria. So it's not that one. Eukaryotes existed before. This is the same statement. It's just written in a different way. Bacteria existed before eukaryotes. C is tracking. D, eukaryotes evolved from archaea. That ain't even in the discussion. C. Ruled them all out. Looking in a microscope, you see a large cell with several membrane-bound sections. What type of cell could this potentially be? Y'all all wanted to just jump ahead and be like, that's a eukaryote. And you're right, it is a eukaryote. Which of these are eukaryotes? And so we just discussed about how bacteria are eukaryotes. Archaea are also eukaryotes. So they're cells without a nucleus. It's just their membranes are wild, right? Viruses aren't even cells. Can't be that type of cell. It's going to be yeast. So we can get to yeast by process of elimination. But in chapter 5, we'll learn that yeast are fungi and fungi are eukaryotes. And so that should be a quicker recall for you. All right, chapter 3, most missed concepts. I think we've got 2 of them. We might have 3, so don't quote me on that. Selective versus differential media. Selective media selects for certain things to grow and selects for certain things not to grow. What can grow and can't grow? You're selective. What can grow versus what can't grow. If it's a differential media, for whatever grows, you will tell the difference. Again, based on usually color changes like pH sensitive dyes and stuff like that. So whatever byproduct you're making causes a color change, usually. And we'll do a ton of this in the second part of the semester, a ton of differential media, okay? Including mannitol, salt agar, and all of the good stuff. So let's get to some questions. We'll write out our little note, our little cheat note at the side. Selective will be what can grow and what can't. Differential, whatever does grow, we can tell the difference. Difference in what? difference in their biochemical or metabolic differences. We'll put by color change, just as there. That's usually what it is. All right, let's see what our questions say. Which of the following is an appropriate definition of selective media? Well, I wrote a very generic one over here, but we'll see what we can match to. Some things can grow and some things can't. Is selective media that changes colors due to metabolic byproducts? That's differential media. That's not selective. Is selective media that favors the growth of some organisms and inhibits the growth of others? Much more eloquently put than can't grow, but that is the gist of it. Media that selects for organisms which require added nutrients. So we're not selecting for organisms that require added nutrients. Some media that we use that has added nutrients to allow other organisms to grow where they couldn't otherwise would be called enriched. That definition is slightly off with that, right? Because we're not selecting for those with that. Media that kills all bacteria except for one type. That's not really the goal of the selective media either. Okay, it's not the goal to kill everything except for one type. Selective media is much more general term than that. Could media that kills all bacteria except for one type be considered selective? Yes, but not all selective media kills all bacteria. This is where we're getting, right? Where that answer could technically be kind of right, but the best answer for this is B. McConkey agar, it has bile salts. Bile salts doesn't mean salt. It means what's in bile, and you dry out bile and the dry stuff that's left over. That's bile salts. It's not the same thing as sodium chloride, okay? So this is what makes bile bile. Bile, okay? This favors gram-negative bacteria, and it inhibits gram-positives simultaneously. So we have something, bile salts, that favors gram-positives, gram-negatives grow, and gram-positives can't. Well, this sounds familiar, doesn't it? Does this sound like this situation? Selective, can and can't grow, right? The media contains an additive that causes bacterial growth to turn pink if it can break down lactose. Well, this is something else. This is a whole separate thing. The bile salts was selective. But if it has an aspect to it that if it can grow, it might turn the media pink. So this is a situation where we have, if it grows, it has a color change. This is differential, right? So what's the question you've been asking? We've gotten that far. What type of media would this be? Is it selective? It is. Is it differential? It is. It's both. Make sense? We're gonna move on. Next is the growth factors. Remember, growth factors are things that have to be added in order for that organism to be able to grow, right? So those are the things that you add. Enriched media has growth factors added to it, and fastidious organisms need growth factors to live. And so we're going to make a little cheat sheet of that out to the side before we even start answering our questions, okay? We'll do it at the top up here. So growth factor, like a vitamin, means that something that the organism needs to be added to their diet. Because the organism can't, this is driving me crazy. In diet, because the organism can't make it. Okay, that's a growth factor. Enriched media, enriched is the term that we use for the media. This is media that has a growth factor in it. Understand that the food that you eat would be your media, right? All right, and then fastidious is a term that we use for the organism. These are organisms that need growth factors, like us. I'm gonna put GF. Why? To the situation, right? Okay, so which scenario details features of a fastidious organism? We can look back up here, a little cheat sheet up here. What's a fastidious organism? A fastidious organism is an organism that needs growth factors. Let's look through our examples. Which one's fastidious? Bacteria that turn pink when growing on McConkey agar. We go back to what we just saw earlier, which is implying that you would have seen what you saw earlier. McConkey agar doesn't supply a growth factor, but it has differential aspect where if you break down lactose, you might turn it pink. But that doesn't make that a growth factor. It's just a differential agent. So it's not really this one. Cows, which are herbivores. Cows eat grass, they don't eat meat, but that doesn't imply the presence of a growth factor, right? Velociraptors. Velociraptors require lysine in their diets. If you've seen Jurassic Park, okay, it's a lysine contingency plan where they say that the velociraptors can't make lysine. They've been engineered where they can't make lysine, and therefore we have to supply them with lysine. But the fact of the matter is, you can't make lysine. No animal can. So, just like velociraptors, me and you, the velociraptors require lysine to live, but they can't make it for themselves, so they have to have it supplied in their diets. That is a growth factor. Lysine is the growth factor. The velociraptor is a fastidious organism, and then whatever food they were eating that had lysine in it is enriched. Okay. Archaea, they grow in extreme environments, but that doesn't imply a growth factor or fastidious situation. Okay. Which of the following could be considered enriched media? Enriched media, remember, it has growth factors in it. Okay, so let's find one that has a growth factor. McConkey agar that has bile salts that inhibit growth of some things and favor growth of others. That's not an enriched media. We're looking for a growth factor. Media that supplies something that the organism can't make for itself. Simmons citrate agar. This one's a little tricky. Only certain kinds of bacteria can utilize citrate as the sole carbon source. Now, bacteria that can use citrate as their only carbon source, they can also use other regular things like glucose as their carbon source. So don't let this trip you up. This is not the answer. Nutrient agar, a basic medium on which a majority of bacteria can grow. That's a general media, the general use, not enriched media. Blood agar. Blood agar contains nutrients that some bacteria need and can't get from other media like nutrient agar. That's more like our enriched media. All right. Does anybody have any questions about the stuff that we just went over? Okay. So if you guys do have any questions, you can always email me and I will clarify whatever I can. So this was our review over chapters 1 and 3. Next week, same time, same format on Zoom at 4:45 every Thursday. We'll go over whatever that week covered. So remember, we covered chapters 1 and 3 this week, which was mostly on you guys, right? If you're in my in-person class, then next week we will be doing our Kahoots and our activities in much more depth for chapters 2 and 4, and we'll get the ball rolling with our actual in-class activities and engagement with this stuff. And those concept maps that you guys are making, they will help a lot more than you realize if you're actually making them the way that you should be, right? That's the whole point. So hopefully you guys be prepared for that. On Monday or Tuesday of next week, we will do the in-class stuff, Kahoot, followed by real Kahoot breakdown, and I'll be calling on you guys, followed by the activity for chapter 2. And then after we've done chapter 2, then we'll do the same thing for chapter 4. I will record that and post that for my online people. And then, you know, what's going to happen with the labs. We'll do lab stuff too. Any questions about any of the lecture stuff before we go one last time? All right, if that is it, I'm gonna let you guys go. If I'm seeing you in my night class, I'll see you in about 1 hour in the lab. Otherwise, I'll see the rest of you on Monday or Tuesday of next week. Have a good weekend.
Thank you.
Thanks, guys.